Welcome everyone, and thank you for joining. Today we'll be hearing from Dr. Kirsten Bott. Kirsten is currently a postdoc in the Bone Imaging Lab, led by Dr. Stephen Boyd and Dr. Sarah Mansky. Kirsten completed her bachelor's in kinesiology at Brock University, where she studied skeletal muscle fat metabolism. She then went on to complete a master's and PhD, also at Brock University. Her PhD was advised by Dr. Sandra Peters and Dr. Wendy Ward. Her PhD work focused on investigating the effect of inflammation on bone structure and bone mineral density. Now that she is a postdoc here at the U of C, Dr. Bott is interested in assessing muscle and bone loss in ICU patients using novel opportunistic CT analysis. One of her proudest achievements is teaching herself how to perform a systematic review and meta-analysis. In the short term, Kirsten hopes to learn as much as she can about opportunistic CT analyses while at Calgary, and also get in as much skiing and hiking as possible. Her long-term goal is to stay in academia and start her own lab to investigate muscle and bone. So with that, Kirsten, I'll turn it over to you for your presentation, and you can start sharing your screen when you're ready.
Perfect. All right, can you see that okay?
Looks great.
Great, yes, so thank you everyone for the introduction, and thank you everyone for joining the seminar today. So I will be presenting on my doctoral studies, just kind of going through the journey and how I got to U of C. So I realize this is a research talk, but I still wanted to give a little background on myself and incorporate my research with my journey to getting to U of C for my postdoc. So I come from the Niagara region, and as you can see, there are a lot of wineries and vineyards, so that's very nice, and I will be missing that now that I'm out here, but the mountains are quite nice as well. And on a clear day, you can see Toronto across Lake Ontario. Niagara is also a really great area for road biking, so I particularly liked riding up the escarpment and taking a little break overlooking the Niagara River. Another activity I enjoy is running, which is nice in the moderate Niagara climate, but I haven't quite worked up to running in when it's really cold here in Calgary yet. But I did, however, find a Hash House Harriers group here in Calgary. So if you know what an on-on is, you're in the know, but I'm assuming most people don't know. So in short, they are an international running group that started in the 1940s by British expats in Kuala Lumpur. They call themselves beer drinkers with a running problem, so two of my favorite things. And in essence, a hare sets a trail that the hashers have to then go find. And the trail is often through a lot of bushwhacking and trees, as well as rivers and mud. So since I began attending the HPL presentations, I've noticed a strong biomechanics gait analysis group, and I'm just saying there may be a study here for analyzing some hash running. So outside of the Niagara region, I also enjoy skiing, so I'm quite excited to be close to the mountains. And since Ontario has very small hills, I took up snowboarding, or for me, it's more accurately a lot of face planting and sitting on my butt. So hockey has also been a big part of my life, and I stayed in school so long just so I could make sure I could get an intramural t-shirt. And finally, now that I'm here in Calgary, I will be missing our family dog Moxie and will gladly offer any dog sitting services. Now on to the more important stuff. So as Emily's already gone over, I was at Brock University for all 3 degrees, which I have dubbed the trifecta. So it's about a 25-minute drive from Niagara-on-the-Lake as well as Niagara Falls. And the picture on the top right there is our bioscience complex. So during my undergrad is when I met Dr. Sandra Peters and joined her lab for skeletal muscle physiology, focusing on the perilipin proteins involved in fat metabolism. And for my master's, I was again supervised by Dr. Peters, but also began to work with Dr. Wendy Ward, and her focus is on bone. It's not that I like her less, I just wasn't completely sold on the idea of continuing to study bone until my PhD when I was co-supervised by the both of them, which is what I'm going to be presenting on today. So moving on, for my presentation today, the main themes for my research talk are going to include osteoporosis in bone. I'm going to be doing a very general discussion on the influence of inflammation in bone, and more specifically highlighting lipopolysaccharide. And this will be incorporated with the considerations of radiation and study design. And finally, I'd like to briefly discuss what I think is an interesting technique for analyzing bone turnover. So osteoporosis is defined as exacerbated bone loss resulting in low bone mass and deterioration in bone microarchitecture, which ultimately compromises bone strength, increasing the risk of suffering a fragility fracture. And bone loss occurs without symptoms unless one has suffered a fragility fracture, and that's why osteoporosis is called the silent thief quite often. Now, traditionally, osteoporosis has been considered an endocrine disease, particularly with the loss of estrogen post-menopause in females. However, it's becoming increasingly recognized that there is an underlying chronic inflammatory pathophysiology. Additionally, other inflammatory diseases that are associated with osteoporotic fractures include rheumatoid arthritis, Crohn's disease, diabetes, and cardiovascular disease. Now, this may in part be due to other factors, such as alterations in mobility/weight-bearing activity and nutrient malabsorption, but again, there is a reoccurring theme of inflammation. So I'd just like to point out that although osteoporosis is clinically diagnosed as a bone mineral density t-score greater than 2.5 standard deviations below the means, individuals can still suffer fragility fracture and may not meet this cutoff. Therefore, it's not only bone quantity. Bone quantity is not always a predictor of overall bone strength. And since bone quantity is only one part that determines overall bone strength, assessing bone structure is also important to understand potential risks of fragility fractures. So why should we care about bone health? So osteoporosis affects more than 2 million Canadians with a high incident rate of fragility fractures, which can decrease an individual's quality of life and puts a large economic burden on the healthcare system. Therefore, it's important to develop strategies to study bone loss and future mitigation interventions. I just wanted to highlight some studies that are looking at the relationship between pro-inflammatory markers in women and men. So from these studies, I've highlighted IL-6 and TNF-alpha, two of the major pro-inflammatory markers we see associated with low bone mineral density, and as well as in relation to fragility fracture. So in females, they only studied hip fractures, and there was a positive relationship between elevated inflammatory markers and the risk of hip fracture, even after doing multivariate analysis to control for factors such as age, BMI, self-reported health, vitamin D, and estrogen use. Whereas in males, there was a more defined increase with the IL-6 compared to TNF-alpha across skeletal sites, but some of the significance was lost after running this multivariate analysis. So additionally, for both males and females, there's an increase in the number of elevated cytokines, was associated with increased age, BMI, increased frailty, and reduced physical function and overall health status. So now moving on to how we see differences or what happens to alter differences and what we see in structure is bone metabolism. So this is the Coles Notes version of bone metabolism. So alterations in bone structure and bone mineral density is determined by the balance between bone-forming osteoblasts and bone-resorbing osteoclasts. And is the balance between osteoblasts and osteoclast activity that determines whether there is overall bone formation or resorption. And then the third major cell is osteocytes, which are mature osteoblasts that have become trapped in the bone matrix. And it's these three cells that communicate with each other to regulate bone metabolism. So the most important signaling is the release of rankle from both osteoblasts and osteoclasts, which can subsequently bind to the rank receptor found on osteoclasts. And this causes differentiation, activation, and survival of osteoclasts, stimulating its bone resorbing activity. Also released is osteoprogenogen, or OPG, and it's released again from osteoblasts and osteocytes. And this acts as a decoy for rankle. Therefore, rankle does not bind to rank, activating osteoclasts and bone resorbing activity. So bone cell metabolism can be altered by a number of different factors, such as nutrition, exercise, radiation, and as I previously mentioned, in relation to osteoporosis, inflammation. So inflammation serves a really important purpose in host defense. So with acute inflammation, it's in response to injury, pathogens, or irritants, and this type of inflammation typically resolves within a couple of days. So it does serve a very important purpose. However, chronic inflammation is less well defined in terms of cutoff points, but is characterized by chronically elevated pro-inflammatory cytokines, resulting in potential tissue damage. So inflammation has been related to a number of different diseases, as I previously mentioned, and this includes musculoskeletal diseases such as osteoporosis. So chronic systemic inflammation is a common attribute to the normal aging process and is often referred to as inflammaging. And there has been links This has been linked with the underlying inflammatory pathophysiology to gut microbiota. So gut microbiota has become a hot topic in the last 10 years. With just a brief history here, the concept of the gut microbiome was only emerged in the mid-2000s. It was upon completion of mapping the human genome in the early 2000s, researchers had all this equipment and someone decided to test fecal samples for DNA. That's when they discovered there was a whole ecosystem living within our gut. The Human Microbiome Project began in 2007 and was a 5-year study characterizing microbial communities. Although this is an area of research in its infancy, we do know that there's increasing evidence that gut microbiota is implicated in health and disease. It's found to play a significant role in host metabolism and can influence a number of different tissues. So bone metabolism is influenced via the gut microbiota by nutrient absorption, so by the resident microbes, so what nutrients are actually being absorbed for the host, in this case us, and its influence on the immune function and hormone secretion. However, I'd like to focus on a specific component, a lipopolysaccharide or LPS, and it's also known as endotoxin in the literature. So typically the intestinal barrier is highly regulated to stop bacteria and other unwanted factors such as LPS from crossing into the host's systemic circulation. However, we know that with aging, the intestinal barrier can become leaky and more LPS can cross. So with LPS specifically, it can affect bone metabolism, which I'll just go into now. So LPS itself is the primary component of the outer membrane of gram-negative bacteria that can be found in the gut, and it's there to provide structural integrity to the bacteria. In terms of its actual structure, it's composed of an O antigen, which is a repetitive glycopolymer, and it's located in the outermost domain of the LPS molecule. And it is the composition of this O antigen that varies between bacterial strains. There is also a core that's connecting the O antigen, and it's composed of oligosaccharides, and this attaches to lipid A. And lipid A is arguably the most important component. It's responsible for much of the toxicity of gram-negative bacteria, and this component is highly conserved between species. So now under normal circumstances, only small amounts of LPS are able to enter circulation, but if LPS crosses the intestinal barrier into systemic circulation, it elicits a strong immune response from the innate immune system. And in high quantities, it can induce sepsis. So it causes an upregulation of pro-inflammatory cytokines, again, the main ones being IL-1, IL-6, and TNF-alpha. And then just two concepts I'd just like to briefly discuss are LPS tolerance. So both in cell culture and animal models, where in primary LPS, there's a primary LPS challenge. It causes an insensitivity to subsequent LPS challenges lasting for hours or days. So LPS can serve as an important role in identifying pathogens for the host. So it's not necessarily a completely bad thing, as it's how the host is able to recognize subsequent infections. And then lastly, metabolic endotoxemia. This has been demonstrated in humans and is defined as chronically increased plasma LPS concentrations, so typically 2 to 3 times higher than what would be considered normal, but about 10 to 50 times lower than LPS levels seen during sepsis. So we have a concept of LPS potentially increasing its systemic circulation with aging as the gut becomes more leaky, and that pro-inflammatory cytokines are related with the risk of fragility fractures. So in terms of LPS in bone, going back to that metabolism component, so the effects of LPS on bone are twofold. LPS can both directly alter bone metabolism by binding to its receptor, toll-like receptor 4, as well as induce an inflammatory state with the upregulation of pro-inflammatory cytokines, which also favor bone resorption. And overall, this is going to increase the release of rankle and decrease the release of OPG, which would favor bone resorption over bone formation. So moving into how we can use LPS to induce inflammatory bone loss, and then how we can actually look at this. So with the human population, it's very diverse, and there are obviously a number of experimental limits. So largely due to these limitations, we use rodent models to investigate various diseases and develop interventions. So with the rodent models, genetically, there are 2 major classes of laboratory rodents. This includes inbred and outbred. Inbred are genetically homogeneous, and there's very little variation within a pure inbred strain, whereas outbred mice or rodents are bred specifically to maximize genetic diversity and heterozygosity within a population. So rodents also offer an accelerated time course and obviously fewer experimental limitations. However, there are still a number of considerations in study design, and specifically with studying bone, irradiation exposure can influence your results. So moving into how we're actually able to assess bone structure and bone mineral density, in humans, we use DEXA or dual energy absorptiometry. And this is used for the clinical assessment of osteoporosis, which again is defined as a bone mineral density of greater than 2.5 standard deviations below the mean. So with preclinical rodent models, we also can use DEXA, and this gives us a measure of bone quantity measured as aerial bone mineral density. So this is a 2D measure. And then we also have microcomputed tomography. So with, again, specific to rodent models, with the microcomputed tomography, it's giving us a measure not only of bone quantity, measured as volumetric bone mineral density for trabecular bone and tissue mineral density for cortical bone, but it also gives us measures of bone structure. So one of the advantages of micro-CT is the analysis that you have the ability to segment out trabecular and cortical bone and analyze them separately. And this can be important since trabecular bone is much more metabolically active than cortical bone. And again, often we see that individuals that suffer osteoporotic fractures don't actually meet that cutoff of 2.5. So the main trabecular bone outcome is percent bone volume, which would be represented as BV over TV. And this is determined by the trabecular thickness, trabecular number, and then the spacing between the trabeculi or trabecular separation. Now, in terms of cortical bone, the most important measure, again, would be a percent bone volume. In this case, it's represented as total area over cortical area. And then some of the other more important measures would include just total area, cortical thickness, and cortical thickness. And we can also measure perimeter, both as periosteal, which is the outer border, and endosteal, which is the inner border surrounding the marrow cavity. And although micro-CT scans offer more detailed information and are considered the gold standard in determining bone structure, this comes at a cost. So with both DEXA and micro-CT, there is unavoidable radiation exposure, but the higher scanning resolution and longer scan times required for the micro-CT result in a higher radiation dose. So in terms of preclinical models, rats are less susceptible to the effects of ionizing radiation than mice, and since they absorb less radiation due to their larger size. And for the purpose of my studies, I use the CD1 mice, and that will be the focus so the previous research, investigating the effects of ionizing radiation at varied doses and intervals in mice. So some of the considerations for doing in vivo micro-CT scanning would be the species and strain of the animals, the age of the animals, the intervention. So this would be the anticipated magnitude of the effects of the intervention compared to the effects of the radiation dose. And it's a relationship between prioritizing capturing potential critical time points compared to the anticipated intervention. And then for the actual radiation exposure, the amount of radiation per scan, the scanning intervals, as well as the number of scans. So these are some of the considerations that should be taken into the study design. So this is just some previous work from other labs that have actually looked at the effects of the repeated information or irradiation from micro-CT. So I just wanted to go over some of that. So I've represented this just by the age of the animals, and then you can see the time frame between each scan. So we can see here that using a total of 1 week intervals for a total of 5 scans had deleterious effects on the trabecular bone. And then within this, you see the effects of the different radiation doses associated, where the higher radiation dose, although there were 2 week scan intervals for a total of 3 scans, this had a negative effect, but using the lower radiation doses in the younger and older animals did not negatively affect the trabecular bone outcomes. And then lastly, in our lab, we tested the effects of repeated micro-CT scans in both male and female CD1 mice, and we used 8 week intervals for a total of 4 scans. We also tested different radiation exposures, including 222, 261, and 460 microgamma rays, and found no negative effects. So the lower resolution scanning parameters associated with the lower radiation exposure, we found that it was difficult to analyze, and therefore the higher resolution scans associated with the higher radiation exposure were preferable. So moving into some of my studies. So I'd just like to note before going on that my radiation study used the control animals from my LPS intervention study, which I'll be speaking about next. So for this study, I tested the effects of the repeated micro-CT scans, but I used a shorter interval, so 4 weeks between each scan for a total of 4 scans. And I based this on the same scanning parameters that were previously used in our lab, where we showed that there was no negative effect on both trabecular and cortical bone. But we wanted to reduce the time between scans to more closely monitor changes in bone outcomes to potentially capture those critical time points. So in terms of analysis, we would find the landmark of the growth plate and then have an offset for analyzing the trabecular bone. And then with the cortical bone, again, we use the offset from that landmark at the growth plate for our analysis. And in this case, we scanned the right leg each time, and the left leg was scanned only at the end for the tibia bone. So these are sample images from the radiation study. And as you can see visually, there are some slight differences between the trabecular bone for both males and females, and then cortical, not so much. And we can actually see that in graph form here. So for the trabecular bone outcomes, following the repeated irradiation, we found that there was a decrease in the percent bone volume for both males and females. And this was due to a decrease in trabecular thickness. There was no change in trabecular number, and an increase in trabecular separation contributed to that change in percent bone volume. Also in the trabecular bone, we found that there was a decrease in the bone mineral density for the males, but not the females. Now moving into the cortical bone. So with the cortical bone, there was less change. There was less of a magnitude of change in response to the repeated radiation, and we only saw it within the females. So within the females, we saw that there was a decrease in the periosteal perimeter, as well as the total area and marrow area as well. However, with the males, we did not see this difference. So in summary, for both the trabecular and cortical bone, the in vivo micro-CT scans using the 4-week intervals for a total of 4 scans affected the trabecular bone in both males and females, whereas in the cortical bones, it was only females that were affected instead of the males. So moving into using LPS in rodent models for examining inflammatory bone loss. So previous work has used slow release pellets, which are incorporated with LPS, and they examined bone structure outcomes. So I've just selected 3 of the most relevant studies. So the pellets work by incorporating the substance, in this case LPS, into the biodegradable matrix that begins to dissolve once it's subcutaneously implanted. So with this, we can see there's varying dosages and study durations ranging from 4 weeks up to 13 weeks. And then all the analysis was ex vivo. So in terms of the results, for at least the drug study, they looked at the femur and found decreases in trabecular bone, whereas with the next study, they only found changes within the vertebral bone, but not the tibia, again, with focus on the trabecular bone. And then lastly, the vertebra and femur, showing a decrease in trabecular bone outcomes, whereas in all 3 studies, cortical bone was not affected by the LPS. But again, this is a less metabolically active tissue. So we focused on the drug study in terms of establishing my study doses. And this is because the study actually tested different dosages of LPS, but only quantified the differential leukocyte counts via blood smears before moving forward using the 1.33 micrograms of LPS per day. So with these previous studies, to date, there's no studies administering exogenous LPS in analyzing bone and that have measured serum LPS. And again, these bone outcomes were only analyzed ex vivo using both DEXA and micro-CT, at least from among the other studies within this area. So another way of delivering substances, in this case LPS, is with osmotic pumps. And osmotic pumps consist of a semi-permeable membrane and osmotic layer that surround the collapsible reservoir. And it's the system that uses the principle of osmosis by drawing water from the tissue in which it's implanted into the osmotic layer, which then compresses the flexible reservoir, forcing the stored solution out through the flow moderator. And the release rate and delivery duration are determined by the composition of the semi-permeable membrane and released in a controlled manner, and therefore more indicative, hopefully, of chronic low-grade inflammation. However, So with the pellets, some previous research that has investigated pellets versus pumps, at least in terms of 17 or in terms of estrogen release, and this is again in animal models, they have found that the pellets either have a supraphysiological or bolus release at the very beginning, followed by a large decline, or there seems to be some variation in the delivery during the duration of the study. So this suggests that pellets may not be the best way of having a chronically delivering exogenous LPS. So for my study, I used the osmotic pumps, osmotic pumps, and I use 8-week-old male and female CD1 mice, which again are outbred breed of mice. And the LPS I used was 055B5, and I based my dosages on the drug study. So using a placebo, low, mid, and high dose. And with these pumps, it has a release rate of 0.15 microliters per hour and a 45-a-day release duration, which gave a little bit of a buffer in terms of our scanning at the 4-week intervals. So again, we were scanning the right legs at each of these time points using the micro CT, and then replacing the pumps at each of those time points except for the last one. So in terms of the results, so we were able to analyze the serum LPS, and I just want to note that there's a fair bit of difficulty with measuring LPS, and this is largely associated with the assays. So we use the ELISA kit, an ELISA kit to quantify serum LPS, which provides more specificity compared to the more classic limeless amoebocyte lysate assay, or the LAL assay. However, we had abnormal results, or not what we anticipated. We didn't see the stepwise increase in circulating serum LPS between our 4 dosages. So for our analysis, we actually did end up collapsing our 3 LPS dosages, low, mid, and high, and comparing to the placebo. So this wasn't unanticipated in terms of our actual results. In terms of our trabecular and cortical bone measures, using the Mark CT, I'm only going to highlight the main measures for at least trabecular bone here. So percent bone volume and bone mineral density. And again, not that we anticipated this, but we didn't see any differences between the 2 groups for the placebo compared to the LPS, but we did see what is to be anticipated with the animals, which is age-related decline in trabecular bone. And then again, with the cortical bone, we didn't see any differences between the male or the controls and the LPS, but we did see, again, the age-related increase in cortical bone that we anticipate during this time point. So in conclusion for this study, the LPS dosages delivered via the osmotic pumps did not alter trabecular or cortical bone structure or bone mineral density in male and female CD1 mice. So although these results were not what we had hypothesized, my supervisors and myself were still interested in the gut microbiota and for me to gain experience outside of Brock. So I had the opportunity to travel all the way down under to Australia to learn how to analyze gut microbiota. This is going back to that journey component. So at the University of Sydney, I was on a project using nutritional geometric framework, and this was to look at the effects of varying macronutrients on different tissues. So this is a massive study I was really excited to be involved with. It consisted of 21 different diets, 5 different time points, male and female mice, and I learned how to extract fecal DNA, and my goal was to map the microbiota profile of the 1-month time point across the different diets. So what I haven't mentioned is that I flew to Australia at the beginning of February of 2020. So in March of 2020, I think everyone can relate to this.
This is a story all about how my life got flipped, turned upside down.
So if that little audio clip didn't play for you, it's just the theme song for the Fresh Prince of Bel-Air. So coming back to Canada, this led to a systematic review. So this is also a really good way of, it was a research blessing in some ways, since doing another study and comparing the osmotic pumps to the pellets for exogenous LPS delivery wouldn't have been as effective as a systematic review. So the main question at the end of my previous study was why our LPS dosages selected based on the previous research didn't demonstrate the same negative effects on bone that we had anticipated. So the study opened up a lot of questions in reference to establishing an appropriate model for systemic inflammation using the exogenous LPS in rodent models. Oops. So for this systematic review, it was titled Rodent Model of Exogenous Lipopolysaccharide Induced Alterations in Bone Tissue. And within the systematic review, the main question was, does exogenous administration of LPS result in a viable model for actually studying alterations in bone tissue in rodent models? And so we were able to search a number of different databases, and we worked with a librarian in developing our search strategy. So the terms for each concept was LPS, or bone, and rodent mole, and these are linked with ORs, and the concepts within these were linked with, within those, they were linked with ORs, and the concepts, those three there, were linked with AND in terms of our search. Now, in terms of the study screening and selection, so we were able to identify just over 1,500 studies for the abstract screening. And then between myself as well as my supervisors, we were able to move through those and eliminate some of the studies that didn't meet our criteria. So our criteria included that the LPS had to be delivered systemically. It couldn't just be injected directly over bone, often the calvaria or the gingiva. There had to be a control, and there had to be bone outcomes that were analyzed. So those are some of the criteria that we had. So I used the circle risk of bias tool for the animal studies, which is based off the Cochrane risk of bias tool. And I just wanted to kind of give my 2 cents on study design and methods reporting. So obviously there was a lot of unclear answers. And of the studies, I emailed most of the authors just for clarification. Some were more major than others. But of the 110 or so authors I emailed, I only had 2 responses, which was kind of interesting. But what I'm hoping for and where we're moving to is with the ARRIVE guidelines. And these are a set of guidelines for improving experimental design and reporting standards specifically within animal research. So although the guidelines were introduced in 2010 and the majority of these studies were published after this date, I think the research has some catching up to do, and I myself reflect back on some of my projects, and I also don't meet some of those standards. So hopefully as we move forward and we do start to see more systematic reviews being done within animal models, these ARRIVE guidelines will help to increase the power of the systematic reviews and the power of that the results. So in terms of the data I extracted, I also looked at study duration. So upon reviewing the study duration, it became clear that there was a cutoff point of about 2 weeks. So of the included studies, 83% had experimental duration of less than 2 weeks, and 17% of studies used experimental duration of greater than 2 weeks. And it seemed that majority of the short-term studies were using the in vivo component to support cell culture work, whereas these longer term studies were more focused on interventions. So this would be the meta-analysis component, and I know it's really tiny, so I'm just going to focus in on the subtotals and totals. So I was able to actually, based on all the data I extracted and using the cutoff points from short-term and long-term, it was only percent bone volume that had a greater than 5 studies to do the actual meta-analysis. So there was lots of studies, at least for BV over TV and the short-term studies, and we were able to subgroup into skeletal site. So within the lumbar vertebra, as well as the tibia, we see here that LPS had a negative effect on the percent bone volume. And then as a total, there was again a negative effect on the percent bone volume. So very similar within all those categories. And again, something to note with these short-term studies is that they used a more homogeneous study design, whereas the longer-term studies seem to have more discrepancies within their study design. So moving on to the long-term LPS, looking at their percent bone volume, again, one of the most important measures for trabecular bone. We see at the femur, as well as the tibia and lumbar vertebra, that LPS had a negative effect or decreased percent bone volume. And again, that resulted in a total as a whole, a decrease in the percent bone volume between all the different skeletal sites. So in terms of the study design, because that obviously plays an important role in how we see our results, we found that with the 2 shorter term studies, they only use the IP injections. So that would be more of a bolus exposure to the LPS, whereas the longer term studies use both a combination of IP injections and these slow release pellets. Dosage was particularly difficult to estimate. A lot of the times the studies would use a relative dosage. So we were making estimates based off the standard weight of an animal. And this also made it difficult back with my study number 2 for determining what dosage I wanted to use, since there wasn't actual dosages provided. In terms of delivery, again, shorter term versus longer term. And we found greater ranges within the longer term studies compared to the short term. So regardless of study duration or LPS delivery methods, so injections versus the slow release pellets, we found that exogenous LPS was consistently reported to negatively impact trabecular bone structure and bone mineral density, while cortical bone outcomes remained unaltered. So to finish off my talk, I just want to discuss what I think is an interesting method for assessing bone metabolism. So in this study, we use low doses of lithium chloride, which is often used as a medication for mood stabilization to treat bipolar disorder. So this is an interesting project investigating the effects of lithium in bone. And since lithium is a known inhibitor of glycogen synthase kinase 3B or GSK3 beta, this can activate the beta-catenin signaling pathway to induce bone formation. So previous studies had demonstrated the osteogenic effects of lithium using higher doses. Therefore, we wanted to use a more therapeutic dose. And the lithium was administered via the drinking water at 10 milligrams per kilogram per day. And this was based off how much they were drinking. We were able to adjust how much lithium to add. And the method that we used for actually analyzing bones, so instead of taking serum biochemical markers, we were actually able to remove the bones and homogenize them, releasing the proteins, and then using that to Western blot 4. So using this bone homogenization technique, we found that GSK3 beta was inhibited and there was an increase in beta-catenin. And this led to an increase in the expression of OPG with unaltered expression in rankles. So that ultimately led to a significant increase in the ratio of OPG to rankle. So had our rodent model of LPS-induced bone loss worked out, lithium was a potential intervention we could have studied to mediate the effects of the inflammatory bone loss. So what's next now that I'm here at the University of Calgary? So I've really actually come full circle. So my master's work looked at the musculoskeletal structure and function. So this was using in vitro muscle function as well as micro-CT to study the bone structure. And then also with the muscles using some fiber typing as well to look at the cross-sectional areas in the different fiber types and whether there was a shift in fiber type in response to an obesogenic diet and aging. So I can change this to University of Calgary now. And as Emily mentioned at the beginning, I am working with Dr. Sarah Mansky and Dr. Stephen Boyd to look at muscle and bone loss in ICU patients using opportunistic screening. So this is where I'm going. And it's nice to see it come full circle. And I'd just like to acknowledge my supervisors, committee, as well as former lab mates and collaborators. So with that, I will end off.
Thanks, Kirsten.
So sorry, I can't see Zoom, so I'm unable to stop sharing my screen. Oh, perfect.
I got you. Great, thank you so much for your presentation. At this time, I would kindly ask the audience members that if you're comfortable to please turn on your Zoom camera to allow for a better discussion. To ask a question, you can use the raise hand function located under reactions on Zoom. And if you're unable to do so, you can type your question in the chat box and I will read it aloud. Michael, please go ahead.
Kirsten, sorry, thanks for the nice presentation. This inflammation stuff's always really interesting. And the big reason is that I look at it, I think of inflammation in two ways. There's these like obesity related inflammation or chronic disease inflammation. But then there's also this like exercise associated inflammation. And I don't understand like why one type of inflammation is good and post exercise and why another type is bad. And if it's some interaction between like the magnitude of the inflammation and the time duration of the inflammation, or like, is it known what the difference is, or what's the threshold for a negative response?
In terms of it being
bone health.
bad?
Yeah.
Bone health? Well, obviously chronic exposure to inflammatory markers is going to influence the bone metabolism. If you chronically have elevated bone absorption compared to bone formation, you're going to see differences in structure and density as well. But I completely agree because I think it's the IL-6 knockout mice. If you exercise them, they don't see the same benefits. It's like the double-edged sword. So yeah, I don't think we have a good handle on that, myself included. I'd be open to hearing if anyone else has anything to contribute for that.
Cool. I look forward to seeing more. It's really interesting to sort of try to tease out why one is good and one is bad.
Yeah, also, I think just in terms of like what we have in the human research, there's like, I'm completely at fault for it throwing around chronic inflammation. I don't know if, like, because we don't have cutoff points that, you know, above a certain concentration that's circulating this is, chronically, that this is what it is. So I think that also muddles some of it as well.
Thank you.
Gable, please go ahead.
Yeah, hi Kirsten, thank you for your talk today. First of all, I've got you covered for dog sitting. Email me. Okay, I got a big dog for you to help out with. Second, I think the gut microbiota bone connection is really interesting. You mentioned it's in its infancy. I still don't know a lot about it, but I think it's certainly an area for future research. Can you comment on LPS in humans and that relationship, or do we always need to measure poop?
So that's where it gets difficult. So like within the geography of the actual gut, there's differences in the proportions of like gut microbiome and like what's going to be at the small intestines versus the large intestines would be very different. And then obviously when you get down to poop, you're still going to see differences that may not be representative of what's up higher in the gut. So in terms of humans, and sorry, you were asking about humans specifically with bone?
Yeah, and I'm thinking, you know, you mentioned, you know, at menopause, we get a little bit more leakage and, you know, can we measure that with LPS? Is LPS affected?
So LPS is quite difficult to measure. So with the animals, we have these ELISA kits, and I think they're coming out with more human ELISA kits. So the LAL assay, it's what we've been using. So it's based on blue horseshoe crabs, and it's using amoebocytes in their blood that clot with the LPS. So that's like the basis of the assay. So it's not the most accurate. So there is research showing that LPS is associated with different diseases. There's only one paper I have found that's actually associated LPS specifically with bone markers. However, it wasn't the best designed study, and some of the conclusions they made didn't match with their results. So that's why I didn't really talk about that one too much. But yeah, we do know with aging that we do see a leaky gut, and there's different ways of actually measuring if you do have a leaky gut. So yeah, sorry, hopefully that answered your question.
Yeah, thank you.
Brent, go ahead.
Yeah, can you just remind me what the primary differences were between the results that you did the meta-analysis, which shows sort of this negative effect of LPS for trabecular bone regions, but then the data you presented in the beginning were unable to find those same things. So I forget, you know, if it was the difference in the animal model or the duration or how it was delivered, but maybe if you could just comment on that.
Yeah, so I chalked that up largely to the osmotic pumps compared to the pellets. So this is one of my frustrating questions that keeps me up at night is why didn't my study work the way I anticipated? So I think that what we've come up with through our discussions through my doctoral studies was that the pumps probably released at such a consistent release rate that the body was able to neutralize it too quickly. So looking back, I would have liked to have done more analysis to see if there was an upregulation in some of the enzymes that would be neutralizing the LPS.
Okay, and then it's interesting to me that you know, you see these changes in trabecular bone, but you don't see them in cortical bone. And we like to say that, oh, trabecular bone is much more metabolically active, but in fact, that's not really true, especially if you look at the endosteal regions of cortical bone and especially in the epiphyseal regions of the joints. And so I'm just wondering, when people conduct these micro CT analyses, do they look at cortical and trabecular bone in the same regions, or do they look at a cortical region in the shaft and a trabecular region, you know, in the epiphysis?
So I know for mine, I did it trabecular bone in the epiphysis and then the cortical bone in the diaphysis. But I know for a lot of at least the clinical stuff that I will be working with, you do segment that out within the same region. So that is a really good point, because I think part of the problem with the micro CT and the size of the mouse bones, from my understanding, is that you don't get a very strong border at the epiphyseal line. So if you looked back at some of the images, you don't really get a clear cut border. So I think it makes it more difficult for the analysis, particularly for like cortical thickness. So
yeah, 100%. I can see that. That makes sense. But yeah, I think it would be really good to look at the changes in bone in the same region, because there is a huge discrepancy between the shaft and the epiphyseal regions. And then the other question I had was, how good of a control, and this isn't necessarily relevant to your work, but just in general, how good of a control is a contralateral limb after you expose one side to ionizing radiation? So, you break a bone on one side, you see systemic losses on the other, you inject Botox on one side, you see systemic bone loss on the other side. And I just wondering if you see similar stuff when you irradiate one side of a limb.
So ideally, we would have had a true control that was only scanned at the very end. I guess the nice part about using the contralateral leg is that it's its own internal control. Now we do move the leg outside of the scanning field by taping it up. But yes, they would most likely get some radiation still, but a fair bit less.
I'm not even referring to the radiation per se, that, the effective radiation that the other limb gets. But I'm just wondering, even if you were 100% able to protect that limb from the irradiation, would you expect the increase in remodeling that you get from the irradiated side to somehow leak over to the right side? That's where I think that would be an interesting study, something to confirm, because it is something that you see with things like Botox treatment and stuff like that. You do get this sort of limb crosstalk, which is quite interesting.
Yeah, I don't know, but I can see your point, because it's essentially affecting the metabolism component, but does that also affect the system? Could be systemic kind of thing. Is that what you're getting at?
Yeah, I'm just curious. Yeah.
Yeah, no, I don't have an answer, and I haven't come across a study that's done, at least not to my knowledge, a true control. It's largely contralateral. Contralateral.
Okay. Yeah. Thanks.
Walter, go ahead.
Great. Thanks very much for the talk. You reminded me of an old friend, the late Cy Frank, who for many years talked a lot about trying to distinguish between the effects of inflammation and the effects of mechanics on the development of knee joint osteoarthritis, and came up with some clever models in trying to really see what does the one do and what does the other do. And so your talk reminded me a little bit of that. So it's kind of nice to see kind of people following up on that inflammation story. The question that I had, well, one of the questions that I had is, in your literature review and your own research, did you ever try to induce LPS, increases in LPS naturally, rather than injecting it or transporting it into the system somehow? And do you think there's a difference between when animals or people have LPS naturally high due to some condition, let's say obesity, compared to when you administer a tablet or somehow exogenous?
So I didn't try doing it naturally. So we really wanted to focus in on just the LPS and inducing that inflammation. So that's the reason we hadn't done that. But there's certainly, with those obese rodent models, increases in circulating LPS for people that have measured it. So part of prior to not in developing my doctoral studies, the plan was that we established this LPS dose. We really thought the mid dose was going to be enough, because that's what Billy Strokitz. So we thought the higher dose was really a bit of an overshoot, but glad we included it. And the plan was to actually look at the gut microbiota to see if there's a crosstalk. So we often talk about the gut microbiota talking to the rest of the body, but how much does the body talk back to the gut microbiota was one of kind of the main questions. At least with that. So that's why we use the exogenous LPS. And then obviously it's more control over that as well, versus inducing it with something like diet or using a genetically modified rodent model.
And so if you do that, and when you do that, when you then measure other inflammatory markers, are they completely normal and you just really upregulate the LPS and everything else stays normal?
So that was one of the I would assume if it had worked properly, yes, we would have seen increase in pro-inflammatory markers. Unfortunately, with the amount of serum we were required to do the LPS, that's what we prioritized. We weren't actually able to measure other markers for either bone markers or inflammatory markers. So unfortunately, we weren't able to look back to confirm that within my study.
So maybe just as an interesting aside, we have this high-fat sucrose-induced obesity model in the rats and the Sprague-Dawley rats, and the LPS levels, serum levels, approximately double after a little while when they become bigger compared to control, and then we kind of see what you are reporting. We see very distinct lesions in the area of the subchondral bone, and then the joint seems to collapse. If you wait long enough, then the joint seems to collapse over these areas of lesions. And so you don't see the regular osteoarthritis, with osteophytes and the cartilage degeneration, but it seems to be a collapse of the bone first, and then other bad things happen. So that's very, yeah.
With those models, like you're also like largely affecting their metabolism as well.
Yes, they do.
So that's part of the reason. I think that's super interesting, but it's part of the reason we did choose to go with the exogenous LPS.
Yeah. Everything's upregulated, all inflammatory markers, not only systemically in the blood, but also locally. We measure in the synovial fluid, and so that happens as well. And the only other thing, I guess, more as a comment, since you're interested in the gut microbiota, you might have heard about Raylene Reimer and other people at the University of Calgary who, I've done this for, she probably was one of the very, very first 10, 12 years ago to start doing that and nutrition. And so you might not have to fly to Sydney, but maybe you want to fly to Sydney, but there's some very good people on the microbiome, as you probably know, and specifically on the gut microbiota as it's affected, and the whole leakage of the intestinal lining and all that type of thing. There's a lot of work being done by people in Calgary that you may or may not want to connect.
Yeah, no, I've definitely heard and read some of that stuff, but I do find it quite interesting. I did have to go to Sydney just based on what the scholarship I received. I couldn't stay in Canada, but Sydney also sounded fun.
Yeah, just
Other questions from the audience? Sarah, please go ahead.
Oh, sure. I was going to pop in last, knowing I can ask Kirsten questions anytime, but I'm just thinking back to the presentation of the systematic review and micro CT. How often has that been done in a systematic review using micro CT data as a primary outcome? Because thinking to Brent's question, I always wonder how much consistency there is in how people are approaching their analysis, and especially when you're talking about things like cortical versus trabecular bone, the approaches to just distinguishing those two compartments are very different across the world. And I'm just curious what you encountered when trying to actually compile that in a systematic review.
The short answer would be poor reporting. So often, like some of my questions, as I said to these authors when I was writing to them, were less serious, but often there was no information on how they did the scans or how they did analysis. Like that was pretty consistent across the board, all the way to not reporting the sample size, the LPS serotype used or anything like that, or even the sex of the animals. So going back to your CT point, I can't really say much on the differences in the analysis that was done with the micro CT and if that contributed to some of the differences between the results as well. But I think we're going to start to see, so going into this, I was trying to learn off other previous animal systematic reviews, and there isn't too many out there, but I think it's going to become more common as well, and should, it's just to reduce how much resources we're using and to move forward, hopefully with better answers.
And similarly, I guess you and I had a discussion about this before, but like this different strains of mice have different amounts of bone. And so are you able to incorporate that into the systematic review in some way?
Yep. So each study, because the analysis was using a standardized mean difference, it's just comparing the control group to the LPS group within that study. So that's a nice part, at least with the meta-analysis. Like you're not technically comparing across studies in that sense, but you're taking the delta change that you're seeing between the two groups and then comparing that across studies. So it does account for some of the study design differences, as well as the different strains and the amount of bone they had. So it doesn't matter if you're comparing a rat to a mouse, that obviously would have very different percent, like amounts of bone, trabecular and cortical.
Thanks.
Bob, go ahead.
Hi, Kirsten. The autonomic nervous system has some control over inflammatory responses, and it also has some control over osteoblast and osteoclast activity. And there are, in some disease states, in bone, but also in blood vessels, it's damage to some of the nerve cells that's considered part of the problem. But in these conversations about osteoporosis and the sorts of studies you're doing, I don't hear about the neural side. Can you comment, given that there is a neural side, can you comment why, or give me your opinion as to its influence on what you're doing? I'm just surprised I don't hear about it.
I'll be honest, I don't know much about the neural side in terms of bone metabolism or bone in general. I think just from my experience, we're just really strongly rooted in it being more of an endocrine disease and looking at the metabolism. That obviously you've pointed out that neural component plays a role, but unfortunately I can't really provide much more, but I'd be interested to hear what you have to say.
I'll send you something.
Perfect. Yeah, no, that's great. Thank you. Sorry.
Walter, go ahead.
I was wondering, since you know that LPS is bad, have you thought a step further on if somebody has metabolic syndrome or other diseases that seem to be producing LPS in excess, on how you might offset that?
Offsetting the LPS, I guess.
Yeah.
First would hopefully be able to improve their intestinal barrier, so you don't have that release of LPS. So one way of doing that would be fibers, pretty common, so that you can have short chain fatty acids, which help to maintain that intestinal barrier. So that's one of the ways I would consider doing that. I don't know, haven't looked into whether there's any drugs or anything that would specifically neutralize the LPS aside from the enzymes to neutralize it within the systemic system. But yeah, I guess I go back to more of a nutrient standpoint. And because it's not that like everyone has gram-negative bacteria within their gut, and it's not that it's necessarily bad. As I pointed out, it's actually really important for identifying pathogens for subsequent infections, and it actually can protect the host as well. So it's important that we are able to identify those pathogens.
That's great, yeah. And in the rats with the high-fat sucrose diet and metabolic syndrome, that's exactly what we did. We gave an oligofructose-based high-fiber diet, supplementing the high-fat sucrose. And that actually seemed to regulate the gut microbiota and the LPS. And yes, there is neutralizers of LPS, for example, intestinal alkaline phosphatase. And we have not started, well, I have a CIHR grant that hopefully will get funded where we exactly try to do that, where we try to neutralize the LPS. So we accept that it's there, we accept that the body produces it. And now pharmacologically, if the grant gets funded, we try to neutralize it and then see if that actually rescues the model. Now so there's other pharmaceutical ways of doing that. Yeah.
With the IAP, is that a direct injection into the gut or just systemically, and that's able to get back into the gut to neutralize?
You can inject it, or you can also just give it in the drinking water for the animals. And we have proposed as an initial step that we would try to just give it orally through the food and the drinking water. Yeah, but you can inject it as well.
Okay, so it can get through all the way without being degraded down into the gut.
That's the theory, and that's what some literature seems to suggest. Yeah, but we are new at this. But there are neutralizers of LPS available, and there's some published literature there.
Yeah. Sorry, I hadn't looked at them. I know there's like the enzymes. I didn't realize it would actually been
And I've only
refined.
looked at that. I've only looked at that in the last couple of years, and you know, so we are new at that as well.
But thank you. I think it's fun.
No problem. No problem.
Actually, I did have a question for you as well, Walter. How did you measure the LPS out of curiosity? Was it using an ELISA or using fecal analysis?
ELISA, yeah.
ELISA
and then, yeah, so the gut microbiota, the whole, the different, you know, composition of the gut microbiota we got from the feces, but then we used ELISA for the LPS, yeah.
And serum for the ELISA or?
Serum.
Serum, yeah. That's what I figured.
Yeah.
Other questions from the audience? Okay, if there are no more questions, please join me in thanking Dr. Bott for her presentation today. Thank you all for signing on. Next week, we'll be hearing from Elizabeth Conlife, and her talk title is Robotic Gait Trainers for People Who Cannot Walk. So I hope to see everyone again next week for next week's HPL Seminar.